I call up the panelists and moderators for ecosystem, privacy, and sharing, Natasha Bonhomme, and the rest of the panel, please, and Vanessa Amandro with Danaher.
Thank you, everyone. I'm very excited to be here today. And by the way, congratulations, Nick and Jeremy, for this wonderful event. Today, I'm going to be speaking a little bit about how I imagine the future that might be enabled by newborn sequencing and how Danaher Corporation might be able to help in shaping this future. So looking forward to statements for the presentation. So before I get to starting talking about the future, let me tell you a little bit more about Danaher Corporation. I am pretty sure that some of you might have never heard about us, but I'm pretty confident that you are working with some of our operating companies. Danaher Corporation is a life science and diagnostic leading company. We are currently actually strengthening our position in life sciences and diagnostics. We announced a couple of weeks ago that we are going to separate our environmental and applied science business to become an independent premium public company. And that's really going to help us to focus in maximizing the impact of the life sciences and diagnostic operating companies that we own, and making sure that we can impact on what we want to do, that is helping realize life potential. Our strategy at Danaher Corporation is to apply the power of science and technology to uniquely engineer impact solutions that can really affect human health. And we are doing that by utilizing our amazing talent pool that we have in the organization. We are more than 70,000 employees, together with our capital and our DBS. And as I mentioned before, what we want to do is truly impact and helping to realize life potential. How we think we are going to be impacting this potential is because we want to own high impact problems, and we want to be able to provide key solutions for our customers and for the scientists that are trying to develop drugs. We have focused our strategy in trying to really enable the discovery of many more medicines by creating a very strong human systems R&D organization. We want to really support more medicines getting to cures, so we have developed a very robust strategy on genomic medicines. Obviously, we not always want to have curative treatments for the patients, but we need to be able to give the right patient to the right treatment to the right patient at the right time. So we are right now reshaping and reinforcing our diagnostic platforms, really focusing on early diagnostics and precision medicine. Obviously, we want to not only identify the patients, but we should be able to manufacture drugs at the right time in a cost-efficient manner to be able to treat our patients. And this is something that I'm going to focus on the presentation in the subsequent slides. Underneath everything that we are doing at Danaher Corporation, there is a significant amount of data that we are generating across all our operating companies. And we are utilizing that data to generate many more insights that can help us in optimize all the products that we have. So now let me switch gears a little bit and tell you a story. When Nick invited me to the presentation, he told me, it would be great to see how Danaher can help us in shape this future. So I'm going to tell you a story first, and then I'm going to tell you how I think Danaher can really help in shaping this future. Imagine a future where every single baby is sequenced at birth. I know that I don't need to tell you that to this audience, you guys are the experts, but just for a second, let's imagine that future. When that happens, when that's enabled, and I don't think it's a matter of if, it's going to be a matter of when and how, newborn sequencing could fundamentally change how we manage our health and we treat diseases. And in this future, we will only be able to unlock the value of genomics if we can really understand how to use the data, how to act on the data, and hopefully use population genomics to really get towards precision public health. So thinking a little bit about macro trends for this particular future, let's think for a second about what can happen when this is a reality. A mom will go, and I don't think I have a pointer. Do you have a pointer? That's okay. A mom will go to, an expecting mom will go to a hospital. She will have a baby. This baby will get, whole genome sequencing. There are going to be sophisticated ways, as we were seeing in the panelists and the presentations before, very sophisticated ways to really get this data, do an analysis, and provide this to a physician that will then be able to use the data to provide a care strategy and a monitoring plan to the families and the patients. And what I mean by actionable data, let's imagine about data that can really lead to treatment decisions, but data as well that will unlock novel patients that are eligible for clinical trials. How many times we have heard about pharmaceutical companies not progressing, drug development programs just because they don't know where the patients are. The patients will be readily available now for clinical trials. There is going to be data that will help in family planning and genetic counseling. And there are going to be risk variants that will be used somehow to provide some risk stratification of the patients, surveillance strategies for these patients. There is going to be many more data that we will not know what to do with it. But as we heard this morning, our ability to really consolidate and federate the data, the genomic and the phenotypic data from the patients along the lifetime of those patients is going to continue to unlock new value on the genomics, is going to really help us to prove the clinical efficacy or the clinical validity, if you will, of genomics in providing care to the patients. It's important to notice that in this future, we are going to need to really modernize the way that physicians can provide care to patients. Right now, our systems are not set up to really enable physicians to maximize the power of genomics to provide this care. And obviously, we will need to truly modernize how we develop and deliver drugs for these patients. Really importantly, we need to keep in mind that this potential future scenario is going to significantly shorten time to diagnosis. It's not going to be like it's happening right now, 5 to 7 years to get someone diagnosed. It's going to be probably weeks. And if we are able now to identify the right patient at the right time, what are we going to do now with this actionable information? How are we going to be treating these patients? So I think it's important to focus. From what I heard this morning, and you guys are the experts, there is significant progress in the space, really trying to lead into getting genomic data to make it actionable. But that means we should be able to act on the data. That means we should be able to treat diseases. As we are getting to this future scenario, this actionable data will probably facilitate access to approved and experimental drugs, most likely to common and rare diseases, but still we will face the problem, what do we do with ultra-rare and N-of-1 indications? For this scenario to be fully true, manufacturing should be immediate because the patients are identified at the right time. It should be cost-effective. We should be able to do a smaller scale N-of-1 GMP manufacturing in centralized and decentralized distribution models, depending on the disease, depending on the setting. We should be able to do multi-market launch. Patients will be identified in every single country. What's the point of being waiting? And obviously, still the system will need to be commercially feasible. So these drugs need to be reimbursable. And I think it's something important to keep in mind that, again, actionability on the data will depend on what can we do after we really have those insights. And for the last 40 years, basically, we've seen a boom in the development and discovery of many biologics and genomic medicines that have been proven in some instances in clinical contexts to provide some cure to patients. But this ability as well, this opportunity that we are bringing now to physicians with many more choices for them to really treat their patients comes with a new challenge for the industry, that is the complexity of manufacturing these potential curative treatments. Let's keep in mind that these are multi-component modalities. This is not, it's not an easy type of manufacturing process that we can think about. And it's important that as we are thinking about manufacturing at large and small scale at the right time in a cost-efficient manner, that we keep in mind that not only we need to do the right process, GMP, high-quality products for these patients, but the treatments should have the right payload that is highly selective for their targets, and they need to be delivered on time and efficiently for these patients. So how can Danaher Corporation help in really realizing this future where we are going to enable actionable genomic data and potentially open the door to have the right patient at the right time? Now what we need is the right treatment for these patients. We have been thinking about this for a long time. Danaher Corporation, it's an organization that with our operating companies, we do not develop drugs, we enable drug developers. We are here to enable those that really are leading in developing these medicines for the patients. So for the past year and with some of our organizations, and I'm just going to give you a few examples on the slide about some of our operating companies, not only we have been providing biomanufacturing solutions for drug developers, but we have also realized that we have an expertise thinking about the ecosystem. How do we work together to really accelerate these drugs? We have the expertise on really biomanufacturing and creating the right environments very quickly. So we have gone and creating Flex Factories and QBio, for example, environments where we are partnering now with drug developers. They tell us what they need. We create this biomanufacturing plan for them, and it's flexible. It's really suited to your needs, depending on the modality, the treatment, the target that you are developing. Not only that, with Cytiva and also Ampol Corporation, we have been carefully thinking about how can we accelerate this process. And sorry, and I want to highlight something about this slide as well. For the first Flex Factories, for example, that Cytiva and Ampol Corporation launched, we were able to help our customers to reduce operating expenses, to reduce CapEx, and obviously to reduce time to market. That's really what I'm talking about, working together and making sure that we can all bring the right expertise to the space to accelerate drug development. In terms of cell therapy solutions, to give you another example, and again, I am by no means a commercial person here, but the point I'm trying to make is that we are really thinking about how can we help the field to accelerate drug development. Not only we have individual instrumentation, we are really thinking end-to-end solutions so that we can carry the heavy workload, if you will, of biomanufacturing and optimizing this process. And we are heavily investing in the organization to make sure that, we are doing good. We are already developing these drugs. We know we can do better, we can do faster, we can do cheaper. So how can we get there? Similarly, we are really trying to maximize our workflows for gene therapy and mRNA, again, going from Aldevron, where we can do GMP plasmid, GMP mRNA, and up to fill finish that we can do with one of our companies, Danaher. I don't want to bore you with more workflows, but again, similarly with CRISPR systems, we actually have a booth here today. I invite you all to just have a chat if you are interested, but it's important that, we have been for the last 10 years as well developing end-to-end solutions for CRISPR, and similarly for Aldevron, for plasmid DNA, mRNA, and proteins at the GMP level. So I'm just gonna wrap up right now, just saying that as before I had a slide that says, is the future inevitable? Well, the future is not inevitable. We all know that we are gonna get there. And the thing that really passions me about what I'm doing at Danaher and in working with some of the key leaders in the space, thinking about newborn sequencing, is the potential that newborn sequencing is going to unlock. And we are not going to be able to realize this future and the value and the power that it carries unless we all work together to enable this future.
My name is Vaughan Witoff. I'm co-founder and chief commercial officer of PetaGene, but don't let that put you off. I'm a scientist and an engineer. The title of this talk is Maintaining Privacy While Giving Access to Genomic Data, something that we're all thinking about at this conference. Alternatively, an alternative title is Demonstrably Achieving Compliance Through Technology Rather Than Relying on Legal Agreements. Now, legal agreements are necessary, but they don't solve this problem by themselves. This is a list. This is a list above the line of customers that, well, a selection of customers that have trusted PetaGene software to compress and encrypt and/or encrypt 4 million human genomes, which is a footprint of more than 40 petabytes of data. Below the line, you can see some of our partners. There are storage companies and producers of fast genomics pipelines and cloud vendors. So these partners have tested our software and have integrated it with some of their products, or at least tested that it works properly with it. So if this technical solution exists or was to exist, what requirements should it have? And spoiler alert, we have a product that does this, which is in use by customers, by some of the customers on the previous slide. So first of all, it needs rigorous encryption. That goes without saying, and it's actually a requirement of GDPR and by implication also HIPAA. It protects the data from breach and leak at rest and in flight, protects data against malicious users and people that shouldn't have access. But what it doesn't do is protect data against people, semi-trusted users that owners of data want to provide access to. And that's where auditing and compliance comes in. The solution must be practical, right? It needs to work with existing pipelines, containers, and workflows. No one wants to adopt a new file format. No one wants to get involved with complicated changes and workarounds. It needs to be high performance. It must not slow down your work. Okay. It needs to be transparent. And that's the key to all of PetaGene's technology. Transparency means that the pipeline shouldn't need to be rewritten. People aren't going to want to do that. And further than that, users don't want to deal with decompression steps, and data stewards do not want to deal with managing keys. It needs to interoperate with existing identity management and access control with GA4GH role-based access. So there shouldn't be an additional layer of authentication, as well as being a vector for attack. That's also inconvenient and complicates deployment. It needs to adhere with the minimum necessary requirement and other rules, and I'll talk about that in a minute. It needs to work across local, cloud, hybrid environments, whatever the case may be, and it needs to be flexible in terms of both internal and external data users. All right, so touching on compliance for a minute, compliance requirements. Both HIPAA and GDPR have, HIPAA calls it the minimum necessary requirement for the privacy rule, and GDPR has a data minimization requirement. I will talk about this in another slide, but sharing a whole genome is normally overkill and violates these requirements. At the moment, there's not much else you can do unless you want to, if you want to work with the original data. The other thing that is a requirement of both statutes is the right to be forgotten. That's the way it's called in GDPR. That once consent is withdrawn by an individual, then promptly access should be withdrawn as well for data that's been shared or has been made accessible. Well, that's a problem with cohorts, right? Especially once they've just been distributed. So we'll talk about how that can be dealt with technically as well. So the minimum necessary rule, I've already touched on this, that it's typically less than 0.1% of the genome is necessary. So sharing the whole genome is certainly beyond the minimum necessary. So quite aside from making available or visible more data than is necessary, it also makes de-identification much more of a risk. Right, de-anonymization, I should say, de-anonymization is much more of a risk when more of the data is shared. So, and compliance within an organization is also important, not just sharing with external organizations. Everything that I'm saying is applicable whether the data is going to be added to a federated storage system or shared locally on a local platform or physically shared by distribution. So how is data shared at the moment? Well, there are two main ways. First of all, the data owner deposits into a centrally controlled access repository the data, and then at some predefined time makes available the keys for decryption, or the data is shared by a direct secure transmission to the collaborator. Either way, once the file has been decrypted, there's no monitoring, and the end user is free to make copies and exploit the data beyond the original data use agreement. That's what legal agreements are for, but how can they be practically policed or enforced? So PetaSuite Protect is the name of the product that we released to address all of these issues. We won Best of Show at BioAT World in 2019, and it's being used by clinical and research customers now. It has the following attributes. So it does transparent encryption without requiring a decompression step. Without requiring a decryption step, I should say. And it also does key management so that the data stewards do not have to deal with that. It gives user visibility to only parts of the genome, the parts that the users are meant to be accessing or need to access. It monitors and logs all accesses for deep auditing. It redacts sensitive metadata without deleting it, meaning that BAM headers can be redacted without actually deleting them from the genome. going towards preventing de-anonymization. It complies with HIPAA and GDPR minimum necessary rule, and it can be transparently integrated with other platforms like Nextflow, Cromwell, 7bridges, Docker, and Singularity, all these tools. It uses strongly robust AE 256-bit encryption, so that's FIPS 140-2 compliant. Each file is encrypted with its own key. For BAM files, you have tens of thousands of keys per genome, and so you have fine granularity. It's an encrypt once. So the files are encrypted once, and the same file is used by all users, but they have that different access depending on what they should be accessing. The data, the users use tracked and logged in a tamper-evident cryptographic ledger. Whether the data is local to the data owner or remote, the data owner sees a full log of usage. And it makes it simple to immediately, retrospectively remove access to individual records from cohorts that have already been distributed. Those files just look like blank to the user. Regional encryption for the BAM files, it's tailor-made to meet the minimum necessary rule. It also allows fine-grained access to specific regions for BAM file and redaction of header fields, as I mentioned, reducing the risk of de-anonymization. That's key for a lot of cohort producers. So how does it work? Well, the data is encrypted once, as I mentioned, and then a copy is shared. It's now encrypted, and so therefore it doesn't have to be a secure transmission mechanism. The user of the data, the end user, with a user mode library loaded on their shell, accesses a virtual file, and the library fetches from the key management server, shown on the left at the bottom, which is under the control of the data owner, the keys necessary for decrypting automatically that part of the file. Assuming that they're authenticated. If they're not authenticated for that part of the file, it's returned as a piece of blank data. The data owner has full visibility into the use of the data, and both parties can agree to expand or reduce access at any time. For those people that are interested in the architecture, there's an encryption step on the left, and there's an LD preload user mode library used for the virtual file production and the presentation of the virtual files, which are then, and the library also fetches the keys. The library presents to the POSIX interface these virtual files, and the applications and tools are running on top of that. Right, so why don't I show you quickly how it works. First of all, the data owner, this is a compression and encryption event, but the compression is completely optional. Encrypts the file, shares those files publicly, and the software stores the private keys. The data steward then logs into the KMS, the key management server interface, creates a project, adds team members, and associates the file with that project, creating a cohort. The data steward sets up permissions. They can use predefined regions, including whole chromosomes, or they can define customized regions, which can be very, very granular. They assign access controls. In the meantime, the data user can use the virtual files shown in blue, looking like symbolic links. They can copy the virtual files, process the files, visualize them just the same as before. All they need to know is that they now use the virtual files. And at any time, the data steward can open the project, examine the event logs, can see which users access which parts of which genome with which tools and which options. The full command line is, they can export the reports for audit. They can also revoke or change access at any time live. So here's a summary. I've covered most of this. I won't go over most of it again, except to say, well, I mean, I've covered the first 5 points quite well. I should mention that it's high performance, both in terms of per file and in aggregate. It's integrated with Active Directory and LDAP user authentication, and it does the J4GH role-based access controls. It supports API admin as well as the graphic user interface that I showed, and supports GA4, GHS GET data access.
Hi there. My name is Stavros Papadopoulos. I'm the founder and CEO of TileDB. TileDB is a universal data management platform, which is architected from the ground up to solve two problems. The first is performance. The second is silos. You may ask, okay, there are another 1,000 databases that are targeting performance in silos. There is a catch with TileDB. We address performance in silos for all data, any kind of data, in a single platform. So that includes tables, genomic variants, RNA-seq, biomedical imaging, and much more. Thank you.
Hi, everyone. My name is Ankit. I've been asking questions from over there. I'm the genomics lead for the AWS public sector. And it sometimes feel like a PhD candidate when I'm asked to describe what AWS does in healthcare. I'm sure a lot of you relate with it, standing in front of a PhD committee and asked to summarize 7 years of work in 30 minutes. But AWS, especially AWS for healthcare, we work across the entire ecosystem, supporting customers in the public sector, whether it's academic medical organization or quasi-governmental agencies, and then supporting our partners who are building solutions on top of AWS for genomics and for healthcare, and then supporting drug development companies, startups, and all, and in the entire gamut. We utilize the power of the cloud. We have the broadest infrastructure deployment, and in terms of compute and storage, so much different services that we can bring to bear. And yeah, lots of great programs like Genomics England, stuff with the UK Biobank, the Hong Kong project, stuff that's happening in the Europe with GA4GH, and stuff with the NIH, the NCI Genomic Data Commons. So lots going on, and would love to talk about building ecosystems while preserving privacy and security.
So I'm David Bick. I'm at Genomics England, and I work with the newborn screening program. I'm a physician, and I have 30 years clinical practice in the US, and then running whole genome sequencing laboratories in the US for the last 10 years, and joined Genomics England last year to help with their implementing their newborn screening program.
Great, thank you so much. So as I ask some questions, and I know questions will be percolating in this audience, so, but first let's kick off with, I love the fact that the word ecosystem is used in this title. And we've talked about this ecosystem and what the data can do and all of that, but where are we actually going? So what is the ideal ecosystem that you all envision in the roles that you play, and how are you building towards that?
Thank you. So I use the word ecosystem as well during my presentation. I think that it's going to be important to realize that as the world and everything that we are doing, especially in the context of newborn sequencing and personalized medicines and curative treatments, things are getting complicated and really sophisticated. So when I think about ecosystem, I think about how can we all maximize what we are good at in doing in our respective industries, but work together to make sure that we can advance the field.
So at PetaGene, we don't like to predict the future, and we think it's dangerous to predict the ecosystems. So all of our software tools have a transparency layer so that any kind of ecosystems can run on top of it. We have this library which gives the data back to the user in the original format so they don't know the difference, and then they can use that in whatever way they see fit.
So one of the biggest problems we identified a long time ago at TileDB, and even before TileDB when I was at Intel and MIT, where this project started, is that, and this is a problem across domains, it's not only in life sciences. We work a lot with geospatial data, and we see the exact same problem. Each of these spaces is extremely fragmented because the data comes in obscure formats. And unfortunately, especially in life sciences, you take those formats for granted. You get, the variant data in a VCF file, and you say, I'm going to keep it as a VCF file because my tools work on this VCF file. This creates huge fragmentation, and it obstructs a lot both the science and the technology, because there are a lot of performance issues that come up because of these obscure formats. GA4GH is something that hopefully will solve this. I personally, and at TileDB, we love APIs. We don't want to buy into a specific format. We have ours because we believe we're building a better format, but it's not the format we're promoting. We're promoting standardized APIs so that multiple different tools and from multiple different teams can be built and shared with the community.
So sort of thinking about, people talk about ecosystems, and, from my biology days, I had a biology degree, like, who's the apex predator? So maybe instead of thinking about an apex predator, maybe we should think about the apex user. And for us at Genomics England, I think the apex user, at least in the newborn program where I work, the apex user is mom and dad, right? They have just had a child, and they expect Genomics England and the National Health Service to, if we're gonna, and we're doing this as a pilot program, you're gonna hear more about that tomorrow, they expect us to take their child's sample, get it sequenced, and get the information to their doctor so that a doctor can call them up and let them know that, yes, their child appears to have a genetic condition, but it's treatable. And they expect, that apex user expects the data to flow completely seamlessly from the time that they sign up, right? Because we're going to have people sign up in the 3rd trimester, to the time that they show up in the delivery room, that the doctors there know to get that sample, for the sample to be obtained, to get into a kit, to get to the laboratory, to get sequenced, and for that data to flow. And so it's not just about the genomic data, it's all of those steps, like from the mom's name to the child's name, to all of that. They expect that to flow perfectly seamlessly and perfectly securely. And so when I'm hearing what you all are doing, your job, in my opinion, is to make the apex user happy. That's my opinion.
Yeah, thanks, Dr. Beck. That was great. And you sort of took some words away from me. And at AWS, we don't like the word ecosystems because, like you correctly said, that sort of still contains you a little bit and tries to define stuff. We have a great philosophy at AWS, especially when we're trying to solve problems. It's called the working backwards philosophy. And it's sort of what you were trying to say is try to identify that end user, the end benefit gainer for a particular problem, and try to work backwards from there. What do we need to do to make that happen? In this particular case, when you're talking about newborn sequencing, it's about the data flowing seamlessly while keeping security in place, while maintaining the privacy of the individual, giving that individual or his guardians, the mom and dads, the ability to choose how to share the data, who to share the data with, and when to basically pull it out. And then supporting how that data now needs to be analyzed and what kind of insights you can generate. And we at AWS, we support all of these functions, all of the partners that are building this and trying to bring together entities to be able to talk to each other. So I don't like to call ecosystems. I like to call a global data mesh, a federated connection of all of the entities that are sort of talking to each other, because that's when true precision medicine starts to happen, right? Especially in the rare disease space. I mean, you can build sorry to take an example, but you can build a whole Genomics England program. But when you start talking about rare diseases, you might still not find enough individuals to get the right signal. And it's only when you start to look at these things from a global scale that you get that power come through. So that's what my vision or our vision is, to help build that.
great, thank you. I may still use ecosystem, but I think we all know what we're talking about. Just because that's what's stuck in my head now. You talk about this global approach, but when we talk about privacy and sharing, this is not a new issue, right? It almost seems like we're building new technologies and new ways of approaching, but the problem is old and just becoming more and more complicated. So from your different viewpoints, where are we on solving the issues between privacy and sharing? Is there one that's going to trump the other? Will we get to a point where at the end of the day, we see the benefit of sharing and that just is what we have to do and that's what we will focus on? Will we say, nope, at the end of the day, it's about each person's privacy and them being able to keep, lock, and manage their data? Is it something in the middle, or are we just rehashing the same question over and over again? I'll start over there.
I mean, in some sense, we are rehashing the question, right? Because we have been asking this question for 50 years now, right? Where exactly is genomics, and how do you tackle it? As bad as the pandemic has been, right? What it did show us is that when you lower some of those barriers on sharing of the data, how you can quickly go from seeing a problem to building a vaccine, for example, to be able to track a disease through the progression with variants and everything, and at a country level. And I think there is an awareness situation that we need to solve. We need to make the people aware that newborn sequencing could help alleviate the challenges in healthcare. And then at the same time, come through in that middle in which you give the end user, the patient, control over their data, how it's shared, how it's stored, who's working on it, give them the full picture of it, while allowing research to happen. So the answer is in the middle, but it starts off with improving the awareness about what genomics is and what it can bring to an individual.
Yeah. Yep. So so parents, if their child has a condition, they want someone to do something about it, right? And parents, it turns out, are by and large quite altruistic when it comes to genetic conditions. I've taken care of lots of children with genetic disorders, and parents specifically ask me, they say, please, David, we would like our data shared. That they come to me without me even asking. In the newborn setting, we need to think about it a little bit differently than the usual rare disease, because these are going to be families who don't have a phenotype and who, for the most part, will have a negative result in a newborn screen. And yet, our experience so far in talking to families, just to try and think about how to move the program forward, are interested in research. They do recognize that the information in their own families, even if their child is well, could help find new diseases and potentially help develop new treatments, cures, if you will. So there is this desire to help their own children and desire to help other children more broadly. And that really is an aspect of what parents do. And I would venture that other people in this room who have children feel much the same way about how they might be willing to share their child's data. And so that creates certain kinds of scenarios in whole genome sequencing in a newborn program, which are going to be very tricky in this space. So, for example, you could imagine a scenario where you screen for 200 genes, but then the next day, because the program will be iterative, you add another gene. What's the way to go back to? Should there be a way to go back to those patients who'd already been screened and look for that condition. How will families feel about having potentially someone go back to them? What kind of choices should they have? Because then they're going to very quickly realize you know a lot more potentially about their child than they do, right? Because if you're only screening for 200 conditions, somewhere back in that data could be BRCA1, right? Like a BRCA gene or something like that. And so this notion of sort of privacy and yet public good is going to be a big challenge in the newborn program. And so that's kind of what we think about, what we're trying to sort out in this space.
I see three aspects to privacy and sharing when it comes to data. One is the consent, everything that the parents need to do, like pretty much what you were saying, and everything that has to do with whether I give the consent for my baby to be sequenced and I surrender forever to you the genome. The second one is regulatory, which is going to happen at some point if this takes off. Like I'm pretty sure regulations are going to start piling up. And the third is the technological. The only aspect I can speak about is the third one. It's the technological aspect. So very quickly, I can tell you that we got you covered there. Not just the TileDB, but in general, that aspect has been solved for many years. Especially in the database sector. I mean, in life sciences, again, all your work revolves around files, and therefore you're finding yourselves reinventing stuff that we built in databases 50 years ago. Once you start encompassing and embracing databases, privacy, data sharing, global scale data sharing, these are solved problems. So the first two aspects, I leave to you guys. You're definitely the experts.
So you're saying it's solved, but I would say most people would not think it's solved. So is it a messaging issue? Is it that our media is scaring us? Or is it that it is solved at a small scale? Or I think maybe elaborate a bit on that.
And thank you for the opportunity. I try to be as concise as possible.
No, that's okay.
Just enough to intrigue you so that you...
I was intrigued. You saw. I was like, tell me more.
All right. So let's talk a little bit about databases. Databases are simpler if you go back several decades ago when everything was tables and simple. There is technology there where the data gets encrypted by default. This is part of the database system. The database system does that. There are access policies. There are role-based access policies. Who gets access, when, everything gets logged. So whenever you ask a query, that query gets logged. So if you're being audited, you can present the logs to the auditor. So from a database perspective, this has been done a long time ago, and I can become even more sophisticated here. In terms of privacy, there are techniques like searchable encryption. For privacy, there is differential privacy. There are techniques, they incur extra costs, like there is a cost issue there, performance and cost issue. But these problems are very well defined in both the database and the cryptography world. Now, in genomics, for example, the problem is the resistance of the domain embracing databases. Check out most of the solutions here. They're working off of files. This is a file, a BAM file, a CRUM file. It's a file. Well, in that case, you need to come up with encryption. You need to come up with access policies. You need to come up with this planet scale sharing system so that, you're talking about federated access. I don't remember in the literature of databases ever ask you the database people using federated. Of course it's federated. It's a database. It's a database system. Of course it is federated. You have root access to install the system and run it, and then you have roles to access the data. So it is a little bit of a fusion of messaging, plus the fact that we tend, when we don't speak to the relevant people, we tend to reinvent stuff that have been solved in the past. Did I cover you?
Sure. We'll see. Any questions around the, I see.
So I'm Tina Erf. I work at NIH at NCATS, and I've spent a lot of time in the newborn screening field with some of my friends here. The question I have is, when it comes to privacy and data sharing, you gave us 4 lovely categories and you said, hey, I've got one of these areas covered. I leave this to the rest of you. And to me, that's a little horrifying in that, it keeps us in silos in the sense that, I'll take care of the technology or the privacy part, somebody else take care of the other parts. And my question to the panel is, how much are you as individual companies taking care of your own areas, communicating with other groups? I know Genome England, you have to talk to everybody. And I love the fact that you brought up that you're talking to the patients, and at the end of the day, that's why we're all here and why we're doing it. So I guess my question is, how much are you interacting with other parts of, and I'm going to say ecosystem, even though we're not supposed to say ecosystem, how much of the environment and other groups are you interacting with to help make things seamless? And how much are you including the patients and patient advocacy groups in these very early stages? Because it's like Natasha was saying, it's like, is this a messaging system? Because people don't understand and people aren't going to accept, it's a perfectly safe system, so just trust me on this. It's like, who needs to do the education and
Can I
such?
start on that?
So
Can I put it a slightly different way? It's not that I don't think there was a suggestion that these problems are separate and we're looking at one of them. We certainly, when we create solutions, technical solutions, we certainly are very engaged with the issues and at the different levels and the reasons that we're going to be. The reasons we come up with these technological solutions is because of the human issues with the other things. It's not that we're not engaged. We're not experts in consent systems and what patients might think about giving consent and what their expectations about the use of genomic data and the outcomes. We're not experts in those things, but we do see potential reluctance in giving consent because of the lack of technical guarantees or technical solutions, and so there is a lack of trust. Potentially, there can be, and then when things do go wrong, it undermines the development of that trust. So I think as companies, tech companies, we're trying to give solutions which will allow the development of trust because the technological stuff is covered. I think that's probably what was meant.
Do you bring the experts in those different fields to talk to you when you are developing these things so that can be taken into account as you build things? Well,
I mean, we're here at this conference listening to what people think, but also we read the literature. Yeah, I mean, we're scientists and we're reading not just the technical literature, but the other literature which is talking about the issues you've talked about. So we educate ourselves about, we don't just base our assumptions on preconceptions, but in the end, we're trying to give robust technical solutions so that you guys can deal with the education of the patients. Maybe.
And if I would just add to that, so I mean, some of the technological challenges, as my colleagues were saying, have been solved. Their application to healthcare is what we are talking about here. That's not, I would say, not been solved to the full extent. And I didn't fully explain our working backwards philosophy, right? I alluded to it a little bit, and you can probably Google it and get much better answers. But the working backwards philosophy says we are working with our customers, right? I mean, I'm sure everybody has shopped at Amazon and talks about customer service and everything and our customer obsession. We identify when our end customers, our advocacy groups, healthcare organizations, they have a challenge. Then we start to work backwards from there to identify what the root challenges and what technological solutions need to be built. But then it doesn't stop there. We then start to talk to more folks that are having the same kind of challenges. There is a statistic out there that says that 90% plus of things that AWS or Amazon builds has been built with this philosophy in mind. So yeah, we are talking with patient advocacy groups. We are talking with research organizations. We are talking with healthcare organizations. And we are trying to understand, other than infrastructure, other than database services, other than storage services, compute, what else needs to be built? And I would love to talk to you about, I mean, I'm sure we are having conversations with NCATS as well, but this is what we are here for today. I mean, I was pleasantly surprised that there's 15 plus presentations tomorrow about various initiatives in newborn sequencing. And I would love to talk to all of you about what kind of challenges you're facing. I understand privacy, I understand ethics. I'd love to see where AWS and our suite of partners can sort of help out.
Yeah, and I would like to add one comment from a personal perspective, and then the Danaher, what we are doing with this. I'm a mom. I have twins. When I was pregnant in my third trimester, actually at the Brigham, they were asking me if I wanted to enroll in a clinical trial, something like that. I don't remember honestly, but it was about doing whole genome sequencing for me and my babies, and I enrolled. But then as I was trying to learn more about, okay, what are we going to be doing with the data, there was a clear path. I totally, withdraw my approval. And I think that we need to keep in mind, we keep talking and thinking about genomics and data and federation and what do we do with the data. The regular population, they are not geneticists. What they want to do is what's coming next after you guys tell me I have something. And that's where at Danaher, we are working with patients. We are really trying to build and think about, not trying to fit the current present into the future of tomorrow. It's like the future of tomorrow has nothing to do with the way that we are operating right now. From a healthcare perspective and drug development perspective, we are working with patients to really understand, and with physicians and hospital systems, what needs to be true for us to be able to enable this. And what patients want are solutions. They will really give us all their data. First of all, if the data is controlled, if the data use doesn't create health disparities, now you might have some issues because you are not genetically perfect. And if they know that they are really providing a bigger help to everyone, and this is the right time to do this. We just came out of the COVID time, where we know that working together, that's the way to really get medicines, to provide impact. So again, let's never forget the patient perspective. It is not about the data, it's what we can do with the data once there is a problem.
I believe we have 2 questions. I think we have enough time for that. So one here.
Thanks. I just wanted to ask, I fear that I should know the answer to this question, but I don't. In a tangible way, if a parent said to me, what are the risks with privacy for this data? I'm just wondering from the panel what kinds of things we should be worried about. So if someone breaches my credit card data, they might buy a car with it. But what are the actual tangible things that could happen with this data that we should maybe be alerting parents to or be able to answer questions about?
Well, I mean, so re-identification, de-anonymization means that if they have a fragment of the DNA or a large amount of the DNA, they can then associate that with your child, with that individual. And who knows what can happen after that. But the point is that now they have the DNA of that person and they know who it is, and they shouldn't. So that's what people worry about.
Yeah. So one comment that goes back to the previous question as well. The way we do it as technologists and as a company is that we collaborate specifically with you guys. We collaborate with Stephen and Rady, for example, in beginnings, and we have constant discussions around consent, regulations, and what kind of privacy and security we're offering. It goes way beyond just a breach. It is my responsibility as a technologist to advise Stephen that, you know what, even if you query statistics from the population and you publish the statistics of the population, it may go back to identifying a single individual in the population. This is what the differential privacy sector is doing. So by partnering, with technologies that have solved these problems in the past, at least you can know exactly what kind of security and privacy the entire system, the entire initiative can give you. And that's why I mentioned that as a technologist, that's what I can give to you, but I need to talk to you and you need to explain to me what is a parent afraid of? Like what kind of a breach, what kind of information that can be leaked in the public is important for the parent, so that I can tell you whether we can do it or whether we can work in the future in order to protect this kind of information.
Our last question.
Hi, I'm Amy Gaviglio. I'm a genetic counselor and a consultant for public health newborn screening programs. And so one thing I've noticed in these talks that has been interesting is that I think we're often using the terms newborn screening and newborn sequencing interchangeably. And that gives me a little bit of pause, because I think in at least in the US, newborn screening programs are public health programs, but probably most importantly to this discussion, they're mandated programs. And what we've really been talking about is consented newborn sequencing. So my question is twofold. One, in the spirit of potentially providing access to everyone to the genomic sequencing, do you ever see this becoming mandated? And if so, how does a mandated system change our discussion around data privacy and data sharing?
That's the perfect question. Thank you. Because so the first step is, does this make sense to moms and dads, right? So that's the first thing. And if it makes sense to them, then it's the people in this room have to sort out the problem that you just said. We should not make our problems the patient's problems, right? So that's always the first rule, right? So if the families think this is a good idea, then we have to sort out exactly the question you're saying. I don't think there's sort of I don't have like an obvious solution to this. And I also definitely think you made a very good point, and a very important point, is that newborn screening as it is today is not newborn sequencing. And so the programs you're going to hear about tomorrow are going to be this first effort to engage with families and get their permission to get their whole genomes and put them in a box. And in that box, we're going to find probably less than 1 in 200 will have one of the, because if you have 200 or 300 conditions on this genome panel that you're going to do, it's going to be less than 1 in 200 will have a positive result. So initially, on the day that you call these people up, hopefully a couple of weeks after birth, that's our kind of plan. Now you've got these people's genomes in a box. Now you need to talk to those families who actually, not theoretically, whose actual genomes are in an actual box somewhere, and ask them what we should be doing next. Because we can ask people theoretically all day about what they think they might do, but it's only when you have real families with real genomes who are really being stored somewhere, then you have the right people to ask what we should be doing. I know that's really not much of an answer.
And I want to add a perspective about this, and I don't think it's going to be all or none, right? In the newborn panel, there is a reason why it's been built the way it's been built, adding new markers when there has been a treatment available or a clinical trial undergoing, like happened with cystic fibrosis, and I happened to work at Vertex for 4 years. So I think that when we are thinking about how to use the data from newborn sequencing to make it mandatory in any panel, as long as we keep identifying mutations that then have treatment available, that might be the way to get there. I don't think we should be thinking about all until we don't understand what to do with everything. We cannot make it mandatory. At the moment that we start generating and validating actionable data, there has to be a way that we can start leveraging and really encouraging people to use this.
Great, thank you.
I would just add one little thought to that. So, and so in the US, and especially in some of the developed world, mandate is a loaded word, right? As soon as we call something mandate, people have a reaction to it. So instead of calling it a mandate, we have to move as a community to a place where newborn sequencing should be as value providing as wearing a helmet while you're riding a bicycle. So we should be showing those success stories. We should be talking about the value that it actually brings to an individual. It should be as simple as people doing after they turn 40, going for their colon exams, for example, right? We have to show enough addition to the value that it becomes a natural thing to do. That's when we truly will come to that place. I mean, mandates sometimes, and especially over the last 2 years, have shown us just get a reaction from people.
Well, I think this will be a very good conversation to continue at the reception later today. I'd like to thank our panelists and for the great questions, and we will move on to the next panel, our closing panel. Thank you.